15-minute or less lecture series, Anatomy and Physiology, Chapter 14, Lymphatic System and Immunity, Part 2. So, as we mentioned last time, we have various lines of defenses. First line of defense to keep things from getting into the body. Second line of defense, always present, always ready to get active for any general pathogen. Then we have the third line of defense. This is the specific immunity. Immunity refers to the response mounted by the body against a specific recognized foreign molecule. These are the antigens on the surface of the pathogens. So antigens, they can be proteins, polysaccharides, glycoproteins, glycolipids. They're on the surface of the pathogen, such as bacteria, viruses, and heck, they're even on the surface of our cells. We have antigens. Remember, blood type A has antigen A on it. Before we are born, we inventory all of our own antigens so that we can recognize our own cells versus not our own cells. And that way we won't attack our own cells. As we know, the lymphocytes are produced in the red bone marrow. The immature T lymphocytes, which is about 70 to 80% of all lymphocytes produced, go to the thymus to mature. Then the B lymphocytes and the T lymphocytes end up in various structures of the lymphatic system, screening for pathogens. So, how do T cells work? Well, we'll start with the cellular immune response. The very first thing that happens is a pathogen, either a macrophage or a neutrophil, engulfs a pathogen, breaks it down, and then places some of those pathogens' antigens on its surface. Sort of think of it as it's saying, hey, everybody, look what I found. These are displayed with the macrophage's own major histocompatibility complex proteins. That way, everyone knows that what's showing this antigen is actually one of our own cells. It's not the actual pathogen itself. Then, a T lymphocyte that is specific to that antigen, and only that antigen, will make contact, cellular contact, with the macrophage displaying the antigen. And if it is a fit, the T lymphocyte becomes active. It becomes an active helper T cell, helper T lymphocyte. So again, if the antigen that the macrophage is displaying is a perfect fit for that helper T cell's one and only antigen it checks for, then they will fit happily, and that'll activate the helper T cell. So after that, the helper T cell will produce chemicals called cytokines. And these cytokines will stimulate that T cell itself to divide multiple times. It'll also stimulate the general activity of white blood cells, leukocytes in the area, because, hey, there's some sort of pathogen there. And it will also stimulate B lymphocytes to function in producing antibodies to also target that same pathogen. So after the cytokines are released, this could potentially stimulate cytotoxic T cells. So the way cytotoxic T cells get activated is first, they find a macrophage that is displaying that antigen for that one specific pathogen. They get a match, a hit. This activates the cytotoxic T cell. This leads to it being ready to be activated by cytokines. The cytokines produced by the helper T cell will then stimulate the cytotoxic T cell to divide many, many times. Then the cytotoxic T cell will go out and look for this antigen being displayed by our own body cells. Basically, it's looking for infected cells. And then it will destroy those infected cells. So it destroys infected cells infected by the pathogen that carries that antigen. Very, very specific. Also, the cytotoxic T cell will divide, and a few of them will become what are called memory T cells. These memory T cells will remember that we've had this kind of infection, and hopefully in the future allow us to rev up a response really, really quickly, so that hopefully we don't have a full-blown infection. And that is the cellular immune response involving cells making contact with cells. Then we have the B cells and the humoral immune response. Here, the B cell needs to become activated. Again, it has special receptors that will bind to the antigen of a specific pathogen. This can be on the pathogen itself, or it can be displayed by a macrophage or neutrophil. Once the B cell has bound the one and only antigen that can activate it, it will be ready to get going. And then all it needs is cytokines that are being released by the helper T cells. So helper T cell cytokines stimulate the activated B cell to then begin to divide and differentiate. So it'll divide many times, differentiate into two different kinds of cells, either plasma cells, which then produce and secrete antibodies against that one pathogen's antigens, And also memory B cells that remember that we've had this infection and will hopefully be able to rev up our specific immune response fast enough to prevent illness in the future. So again, this is a humoral immune response because the B cells don't directly attack the pathogen. Instead, they produce antibodies that enter the fluids and bloodstream and then attack the antigen indirectly. There are 5 main types of antibodies. You have all of them, though, having certain constant areas that are always the same, and then little variable areas that change. This is the part, these variable areas, that are specific to one and only one type of antigen, so that each antibody from a different cell will be targeting a different type of antigen. So we have IgG. This is the most common kind of antibodies. It's found in tissue fluids. It's found in blood plasma. It attacks bacterial cells, viruses, toxins, etc. It can deactivate viruses and toxins. It can make pathogens a target for phagocytosis, a target for complement, putting holes in those bacteria. So if you remember, complement is proteins already found in the blood plasma. They can stimulate inflammation, attract phagocytes, and also cascade out to form holes in the membranes of bacteria, destroying those bacteria. Then we have IgA. IgA is in our secretions by our exocrine glands. So it's found in breast milk, it's found in saliva, tears, gastric juices, etc. It tries to attack pathogens before they get into the body and also pass on temporary immunity to infants. IgM is found in the blood plasma, and it can activate complement, just similar to IgG. It can cause agglutination. As you can see, it has many binding sites on one IgM antibody, and it is the antibody that is produced that ends up causing problems in blood transfusions, so that if you get the wrong blood type in a blood transfusion, it's this antibody that causes them to agglutinate, to clump all together, leading to serious problems. IgD is actually found on the surface of the B lymphocytes. So the receptor that the B lymphocytes use to recognize the antigen that they are opposed to is actually IgD, a type of antibody. And then IgE is also an antibody excreted by exocrine glands, and it also can lead to allergic reactions in the body by stimulating mast cells that then produce lots of histamines and other chemicals that cause the major allergic reaction. So antibodies have 3 main functions. They can attack by binding to the antigens on the surface of the pathogen and causing them to clump up. So again, getting the bacteria to form a big clump by all these antibodies, binding to them, chaining them together, making them easier to find. They can also activate complement. This can lead to opsonization, making it easier for a phagocytic cell to engulf the pathogen. It can lead to chemotaxis, makes it easier for the pathogens to the phagocytic cells to find the pathogens. It can cause inflammation, lead to a lysis of the target cells, etc. And the third way is inflammation. The antibodies can then stimulate changes in the area to lead to inflammation, the increased blood flow, leading to pain, swelling, redness, heat. This, of course, attracts leukocytes, which come in to attack the infection and then isolate the infection so it doesn't spread further. So, our specific immune response. Usually the first time you're exposed to a pathogen, it'll take a while to get your immune response generated, and even then it's not as good as it could get. So it takes a little time for the B cells and T cells to activate, and it's usually a little slower. But the memory cells will be produced. With the memory cells now in your system, if there's a secondary infection, the specific immune response will occur rapidly and lead to a very dramatic attack on these pathogens. And this is what immunity is. We still get infected, but we're able to deal with the infection quick enough that we don't actually suffer any symptoms of the infection. Different classes of immunity. Naturally acquired active immunity is basically what I've been talking about, where your T cells and B cells all get involved in destroying the pathogen. Artificially acquired active immunity is when you use vaccines. You don't actually get infected by the pathogen. Instead, a vaccine, an artificial intervention, exposes you to the antigens so that you build the immune response, build the memory cells to help prevent disease in the future. Artificially acquired passive immunity is where they take antibodies from the bloodstream of some other person or animal, and these antibodies attack specific antigens, and then they can be injected in someone as a medical treatment. For instance, the treatment for snake bites basically are antibodies that attack the toxin the snake injected in the person. And finally, naturally acquired passive immunity is when antibodies, especially IgA, are passed from the mother to the infant during breastfeeding, which is short-lived, but it is important to keep the infant healthy. Allergic reactions are when our immune system responds to things that are not a problem, that are not dangerous. So allergens are not dangerous to our body, but someone with allergies has an excessive immune response to it. This can include things like pollen and mold spores and nuts and milk and eggs and pet dander and all kinds of different things. Uh, the immediate reaction allergy is usually inherited and leads to an overproduction of IgE against that allergen, which then stimulates mast cells. Mast cells then release chemicals that stimulate the allergic response, leading to vasodilation, redness, and swelling, leads to contraction of bronchioles, so it's a little hard to breathe, excess mucus production, pretty much all the things that people have to suffer if they have allergies. Um, Sometimes the allergy mediators flood the system, the body system too much and lead to anaphylactic shock and death, specifically a very severe form of immediate reaction to an allergen. Delayed reaction allergies, basically you need repeated exposures before you begin having allergic responses, inflammatory responses to that allergen. Sometimes it can take 48 hours or longer. And basically, when someone gets, say, a test for various allergens, the idea here is they inject tiny bits of the various allergens into a person's skin, and then if there's any swelling over time, they know that that person is allergic to that substance. Pre-allergy, avoid allergens, duh. You can try treating the symptoms themselves, make them less severe, decongestions, gestants, antihistamines, etc. Mast cell inhibitors, allergy shots. It is possible sometimes to build up a response using IgG antibodies so that they bind the antigen faster than the IgE antibodies. Okay, moving on to transplantation. It turns out that you have to get a good match between a donor and the person receiving the tissue, or else you get tissue rejection. So here, your own tissue can be moved around, that's fine. You get a good matching donor, you should hopefully not have rejection of that tissue, but if it's not a good match, you end up having rejection. The immune system attacks the tissues. So you need a close donor match, which basically means their MHC antigens need to be very similar. So you don't need all genes to match, just the ones that are going to be found on the surface of cells. After a transplant, then immunosuppressive drugs are given to the person to try to reduce the possibility of rejection, because if the organ is rejected, then they have to go through surgery immediately to get it removed and replaced. There are also autoimmune disorders. This is when your own immune system is attacking its own tissues. This could be because the antigens from some pathogen are very similar to antigens on your own cells, leading to accidentally attacking your own cells. It could be by a weird viral infection causing a trigger to attack your own cells. There are a lot of very possibilities, including, say, mutations in the stem cells that produce the blood cells, leading to you starting to attack your own cells. Lots of possibilities. Some examples of autoimmune disorders include lupus, type 1 diabetes, attacking parts of the pancreas, rheumatoid arthritis, attacking the joints, multiple sclerosis, Crohn's disease, psoriasis. There are a lot of autoimmune diseases out there. And some diseases that they may someday find have an autoimmune link.